During the M phase of the cell cycle, each cell will divide into two daughter cells. The process of the cell division involves two main processes. First is the division of the nucleus or mitosis. Secondly, followed by division of the cytoplasm or also known as cytokinesis. During interphase, the DNA of all eukaryotes exists in the form of chromatin, which refers to the thin and invisible structure where the DNA wraps around the histone protein. During prophase, which is the first stage of mitosis, centrosomes, which consist of centrioles and esters, will start to migrate to the opposite pole of the cell. Simultaneously, the chromatin will start to shorten and condense to form a thicker structure called chromosomes. The nuclear membrane will also start to disintegrate and disappear. Next, spindle fibers starts to form from esters of the centrosome. Finally, at the end of prophase, chromosomes are now fully visible, where we can see each chromosome will have two sister chromatids attached at their centromere. In the second stage of mitosis, which is metaphase, both centrosomes will arrive at the opposite pole of the cell. Each chromosome will have spindle fibers attached to its centromere coming from the opposite centrosome. At the end of metaphase, all chromosomes will become aligned at the equator of the cell, known as the metaphase plate. Notice that all chromosomes are aligned individually along the metaphase plate. During anaphase, spindle fibers will start to shorten and pulls the sister chromatids at their centromere towards the opposite direction and causes the centromere to split into half. This causes the sister chromatid to separate and become individual daughter chromosomes. As the spindle fibers continue to shorten, the daughter chromosomes will be pulled away from each other to the opposite direction. When all daughter chromosomes have arrived at the opposite pole, that is the end of anaphase of mitosis. The last stage of mitosis is telophase, where the nuclear envelope will start to reappear, spindle fibers disintegrate, and chromosomes start to uncoil and lengthen and become invisible once again. At the end of telophase, two genetically identical daughter nuclei will be formed. Completion of telophase marks the completion of mitosis, which is the nuclear division. However, this is not the end of the cell division process. The process of cell division will continue with the division of the cytoplasm, which is called cytokinesis. In animal cell, cytokinesis takes place by formation of cleavage furrow. Cleavage furrow is formed due to constriction of rings of microtubules at the equator of the cell, which causes the cell to split into half as the cleavage furrow deepens. Once cytokinesis has completed, two genetically identical daughter cells are produced. That is the end of the M phase or mitotic phase of the cell cycle.